In this video, I'm going to review the process of transcription, which is converting a DNA template into an RNA molecule. Now, there are many different types of RNA molecules that can be transcribed. There can be messenger RNAs, transfer RNAs, ribosomal RNAs, and many others that we're not going to talk about. Now, the key thing here is that there is a lot of DNA within a genome, whether it's an E. coli genome or a eukaryotic genome. Only specific regions of each genome are going to be transcribed, and those regions are called genes. Genes all have a few things in common. They all have a promoter sequence, which indicates where the gene is in the genome, and it's going to allow the assembly of the transcriptional machinery. There's going to be a transcription start site, which we often indicate with an arrow in the direction that transcription will occur. And then there is a termination site where the transcription is going to stop. So we'll break transcription down into initiation, elongation, termination, and processing like we did with DNA replication. So the promoter is where it's at. This is where initiation is going to happen. Regardless of the cell type, the first thing that has to happen is that the RNA polymerase is recruited to the promoter and stabilized in the proper orientation. So this happens in subtly different ways depending on if you are looking at an E. coli or bacterial cell versus a eukaryotic cell. So in the E. coli, the RNA polymerase is bound to a protein called sigma factor. And sigma factor directs the RNA polymerase to promoters and allows it to get settled onto the DNA there. In eukaryotes, first there are a series of proteins called transcription factors, which have to bind to the promoter. And then those transcription factors will recruit the RNA polymerase to the promoter. Okay, so the next thing that has to happen is this whole complex needs to be stabilized. And that happens through the action of RNA polymerase. So RNA polymerase itself can unwind the double-stranded DNA at the transcription start site. The other thing that RNA polymerase can do is start transcribing de novo. This is just a fancy way of saying that the RNA polymerase doesn't need a primer. It can start transcribing from nothing. Now, of course, it does need a template DNA to do the transcription. It just doesn't need a free 3' hydroxyl. Okay, so during elongation now, RNA polymerase will have this little RNA that it started making, again, in the 5' to 3' direction, and it's going to continue to add complementary bases, bases that are complementary to the DNA template. So again, the base pairing rules for DNA RNA are if the DNA template is like this, then you'd have U base pairing to A base pairing to T in the DNA, and then C and G. And that's what the RNA sequence will be. Now, the other important thing to consider is that since RNA polymerase is synthesizing in a 5' to 3' direction, that means that the template DNA has to be running in the 3' to 5' direction. So if this was a little piece of DNA like that, it would have to be 3' to 5' in order for the RNA to be made 5' to 3'. So our RNA polymerase is going to just move along the DNA template until it reaches the termination site. Now, as it goes, it's going to create this little bubble, which is much smaller than a DNA replication bubble. This might only be like 15 to 20 nucleotides here. And in the wake of the RNA polymerase, so behind where the RNA polymerase has already been, the DNA will reform a double-stranded DNA molecule, right? That it'll reanneal, and it can be used again to as template for another RNA polymerase. So as I mentioned in class, termination is actually kind of complicated. There are many different ways that termination can occur in eukaryotes, a couple different ways that usually happens in E. coli. For the most part, the way that we should think about it for the purposes of this course is that there's going to be some kind of terminator sequence that is at the end of the gene. So this terminator sequence is going to be transcribed into RNA, and then that does something to the RNA or recruits something to the RNA that causes the RNA polymerase to fall off. So once that happens, then this mRNA is going to be free to go do its job. So it'll be free like this with a free 3' end and 5' end and 3' end. So if this is an mRNA and we are in a bacterial cell, then that mRNA can be translated right away. In fact, it could be translated while it's getting transcribed because it's happening in the same compartment in the same site in the cytoplasm. If this was a different type of RNA, like a tRNA, then it will fold up and be ready to do its job. Now, if this was an mRNA that was in a eukaryotic cell, it needs to be processed. And there are 3 processing events that occur. First, a 5' cap has to be added. The function of that 5' cap is to provide protection against exonucleases that might try to hydrolyze it. Or another way to say that is to keep the RNA stable. It also is needed to get out of the nucleus, and we call that nuclear export. So in order for it to actually get out of the nucleus, it has to have this cap on it. Finally, it's going to be involved in translation initiation. So in order for an mRNA to be properly translated, it has to have a 5' cap. The second processing that needs to occur is a 3' poly A tail. So for our purposes, it essentially does the same thing. So it protects the 3' end, again, from getting hydrolyzed by exonucleases, keeps the RNA stable. And then it's also involved in getting the RNA out of the nucleus and initiating translation. Okay, and then the third processing event that has to happen is the RNA needs to be spliced. So that means that the introns of the RNA are removed and the exons are spliced together. The ultimate effect of splicing is that the proper coding sequence will be generated. So if we consider this to be our unprocessed mRNA, initially the 5' cap is added, and this is a modified nucleotide. You don't really know the details of that, just know that it's a nucleotide that gets added to the end here. Then once the entire transcript has been made, transcribed, this is when the 3' end is free and the multiple adenines can be added at the end. This N is just representing the number of adenines. It can be anywhere from 50 to 250 or actually even more or less. It just depends on the transcript, but this is sort of the average. So again, there's an enzyme that will bind to the end and then add a whole bunch, covalently link a whole bunch of A's in a row. Okay, then finally in splicing, this intron, for this particular mRNA, it has a single intron, but some RNAs have many introns that have to be spliced out. And the exons are going to be joined together. So we would have the exon sequence from this one and the exon sequence from that one joined together. The poly A tail and the 5' cap are there. And now you have the fully processed RNA. And this is often referred to as a mature mRNA as opposed to an unprocessed mRNA. Okay, so the last thing is if we look at this mature mRNA with all of the exons spliced together, there are going to be some special features to it. So one thing that you will see is that if this is encoding a protein, which all mRNAs do, there needs to be a triplet that is used as the start codon, which is the first codon that will be used by the ribosome to actually make the protein. So that start codon is going to indicate where the protein coding sequence is. So if we were to zoom in and look at the sequence here, it would be an AUG at that start codon. Okay, now there also has to be a stop codon, which indicates where the ribosome is going to stop translating that protein. And so if you zoomed in to the stop codon, you're going to have a stop codon. And if you refer back to your genetic code, you'll see that it could be one of these 3 stop codons, UAA, UAG, or UGA. So all of the RNA sequence between the start codon and the stop codon is going to be the protein coding sequence. So that all of those triplets in between the start and stop codon are going to encode for specific amino acids that are going to be read off when translation occurs. That means that the sequence upstream of that is not translated. So we call that the untranslated region. And it's specifically called the 5' untranslated region, which is kind of a lot to say. So we usually just refer to it as the 5' UTR. And maybe I'll just put a little highlighter to indicate where the UTR is. So then again, this coding sequence would be between the start and stop codon. And now we have another UTR, and that's going to be the 3' UTR. And I'm just going to put the abbreviation. So I didn't really mention this in class, but the UTRs are important for translational regulation. And so they play a role in making sure that this RNA is translated properly and also involved in keeping the RNA stable. Okay, that's it for transcription. Now check out the translation video next.